Hi, everyone. Today we are going to talk about some complex carbohydrates. So monosaccharides are linked to form complex carbohydrates. Because sugars contain hydroxy groups, glycosidic bonds can join one monosaccharide to another. Oligosaccharides are built by the linkage of two or more monosaccharides by all glycosidic bonds. In the disaccharides, maltose, for example, two D-glucose residues are joined by a glycosidic linkage between the alpha-anomeric form of carbon 1 on one sugar and the hydroxyoxygen atom on carbon 4 of the adjacent sugar. So such a linkage is called an alpha-1,4 glycosidic bond. The fact that monosaccharides have multiple hydroxy groups means that many different glycosidic linkages are possible. For example, consider three monosaccharides: glucose, mannose, and galactose. So these molecules can be linked together in the laboratory to form more than 12,000 structures, differing in the order of the monosaccharides. And the hydroxy groups are participating in the glycosidic linkage. So in this section, we will look at some of the most common oligosaccharides found in nature. Sucrose, lactose, and maltose are the common disaccharides. A disaccharide consists of two sugars joined by an O-glycosidic bond. Three abundant disaccharides that we encounter frequently, as we mentioned, remember, is sucrose, lactose, and maltose. So sucrose, also common for like table sugar, is contained commercially from sugar cane or sugar beets. The anomeric carbon atoms of a glucose unit and a fructose unit are joined in these disaccharides. The configuration of this glycosidic linkage is alpha for glucose and beta for fructose. Sucrose can be cleaved into its component monosaccharides by the enzyme sucrase. Lactose, the disaccharide of milk, consists of galactose joined to glucose by a alpha-beta 1,4 glycosidic linkage. So lactose is dehydrolyzed to these monosaccharides by lactase in human beings and by galactosidase in bacteria. In maltose, two glucose units are joined by an alpha-1,4 glycosidic linkage. Maltose comes from the hydrolysis of large polymeric oligocytes such as starch and glycogen, and is in turn hydrolyzed to glucose by maltase. Sucrase, lactase, and maltase are located on the outer surfaces of epithelial cells lining the small intestine. So the cleavage products of sucrose, lactose, and maltose can be further processed to provide energy in the form of ATP. Glycogen and starch are storage forms of glucose. So glucose is an important energy source in virtually all life forms. However, free glucose molecules cannot be stored because in high concentrations, glucose will disturb the osmotic balance of the cell, potentially resulting in cell death. The solution is to store glucose as units in a large polymer, which is not osmotically active. Larger polymer oligosaccharides formed by the linkage of multiple monosaccharides are called polysaccharides and play vital roles in energy storage and in maintaining the structural integrity of an organism. In all of the monosaccharide units in a polysaccharide are the same, So the polymer is called homopolymer. The most common homopolymer in animal cells is glycogen, the storage form of glucose. Glycogen is present in most of our tissues, but is most abundant in muscle and liver. Cellulose, a structural component of plants, is made of chains of glucose. So it is another major polysaccharide of glucose found in plants. And the cellulose serves a structural rather than a nutritional role as an important component of the plant cell wall. Cellulose is among the most abundant organic compounds in the biosphere. Cellulose is an unbranched polymer of glucose residues joined by beta-1,4 linkages. In contrast with the alpha-1,4 linkage seen in starch and glycogen. This simple difference in stereochemistry yields two molecules with very different properties and biological functions. The beta configuration allows cellulose to form very long straight chains. Fibrils are formed by parallel chains that interact with one another through hydrogen bonds, generating a rigid supportive structure. The straight chains formed by beta linkage are optimal for the construction of fibers having a high tensile strength. The alpha-1,4 linkages in glycogen and starch produce a very different molecular archetypes. So like a hollow helix is formed instead of a straight chain. So that's all for today about the polysaccharides, and thank you for your attention.